Hi, you guys. I'm here to walk you through Chapter 3, Cell Metabolism. This is a really big chapter as far as concepts and topics go, a lot of chemistry. So, here we're going to focus on the mitochondria, which is an organelle introduced to us in Chapter 2. And the goal is to make lots and lots of ATP energy from the foods we eat. So you'll see some overlap with nutrition and chemistry, and some of these slides were given to you in the biochem review notes as sort of the prerequisite for biology and chemistry that will be applied directly to this class. So our outline is going to be looking at rates of chemical reactions, so how fast things go over units of time, and enzyme function. Enzymes are very important to controlling metabolism. And then probably in a second lecture, we'll get into glucose oxidation, aerobic respiration, and then what happens when we have more energy than what is used. It goes into storage. So to start off, metabolism is a term that we hear a lot. And as a science person, you should know that metabolism is a collection of many chemical reactions that make products that are used in other reactions. So in the human body, it's extremely efficient and nothing ever goes to waste. Think of metabolism as a chain reaction. The product of one reaction is used as a reactant in the next. Metabolism is either an anabolic process, which is a constructive process and new molecules are assembled, or it's a catabolic process where molecules are broken down into smaller component parts, like what happens during digestion. When you eat food, it's broken down into smaller molecules. So here's our outline for Chapter 3. And the next slide, just want to walk you through a few things in case these abbreviations look sort of foreign. A lot of times when we're in class together, I'm drawing on the board as I'm going through my PowerPoint slides. So as you can see here, in this reaction with A plus B makes C and D. So A and B are on the reactant side. They're added together, and over time, they create some product, C and D. Now, certain factors will make this type of reaction go faster. Metabolic reactions are accelerated or catalyzed by special proteins called enzymes, and factors that will make a reaction go faster tends to be things like concentration, temperature, and if there's any barrier to making product. So the concentration of A and B uh, will have a tendency, if you have a high concentration, it's more likely that you'll get product. If you don't have a lot of A and B around, it may be difficult to make C and D. Temperature is another factor that can affect the speed or rate with which a reaction will push forward. Usually when you heat up a reaction, it goes faster. Energy barriers that must be overcome will be demonstrated as like a hill. The energy that it would require for you to haul a load of cement in a wheelbarrow up a hill is going to require energy. But once you're at the top of the hill, you could roll the wheelbarrow back down without any effort at all. So we'll talk about energy barriers and the presence of enzymes and how these special catalytic proteins will help push push reactions forward. There are three key reactions that are very important, and they're constantly taking place in the human body. The hydrolysis reaction, hydro meaning water, so using water to split the bonds of a chemical by using water. This is a typical catabolic reaction. So if you think about digestion and the breakdown of foods that you eat into smaller particles, hydrolysis is what goes on in the GI tract. Another important reaction is the phosphorylation reaction, where a phosphate group is added to a molecule. This is a common reaction in metabolic processes that involve energy exchange. And we'll come back to this reaction when we talk about the creation of ATP. Another important reaction is the redox reaction. Oxidation reduction reactions are termed redox. They are a key cellular metabolism reaction. These two reactions are always coupled together, and they're important for the understanding of cellular metabolism, which we're going to get into here in just a minute. A substance that gets oxidized is in the process of losing electrons. So think of LEO, Leo, loss of electrons, oxidized. And the opposite happens when a molecule gets reduced. It gains electrons. So GER, gaining electrons reduced. That's my little tip for how to remember what's getting oxidized and what's getting reduced. Leo, the lion, goes GER. Leo for oxidation and GER for reduction. Metabolic reactions provide the body with energy that it needs to do work. Work is usually in the form of movement, and it's expressed as force times distance. Living organisms need a constant input of energy for the three major forms of work. Transport, so moving molecules, like in the case of diffusion. We'll be doing lab number 2 to study osmosis and diffusion. There's also facilitated diffusion and primary and secondary active transport. These are all ways of moving molecules in a cell, usually across the plasma membrane. Mechanical work, locomotion and cytosolic traffic, moving molecules around inside the cell. Usually the cytoskeleton and the microtubules and microfilaments are part of that mechanical work inside the cell. And of course, chemical work, synthesis, including ATP and GTP, which is a close cousin to ATP. So instead of an adenine, there's a guanine. And you should be familiar with A's and G's and T's and C's from our discussion on DNA. Okay, so let's look at the different ways a cell can perform work. And just remember the laws of thermodynamics still apply. There are two forms of energy, kinetic or potential energy. So at the top of the hill, just before you roll the ball down the hill, that's potential energy. Once the ball is set in motion, it becomes kinetic energy. The laws of thermodynamics state that energy cannot be created nor destroyed. It simply changes form. Energy, like molecules, will diffuse from areas of high energy to areas of low energy. Thus, the analogy of using a hill. On the right, we can see some examples of cellular work. These require energy. You can see ATP being put into the system in order for this work to be done. Energy is being converted from one form to another in order to support life. So at the top, we're transporting a molecule from one side of the plasma membrane to another. In the middle, muscle is going from contracted to relaxed and back and forth at the expense of ATP. And at the bottom is chemical work. So this looks like a phosphorylation and then a dephosphorylation process. So X and Y are joined together as the product. So these are all different examples and should be recognized as energy being converted from one form or another in order to support life. Now, enzymes are special proteins with catalytic ability, which means they can catalyze a reaction to make it go in the forward direction much faster and at a constant temperature, like our internal set point. Our temperature cannot be increased or decreased by very much. We stay at 37 degrees C. But some reactions may not go very well at that temperature. A lot of times in the lab, we heat something up in order for it to go faster. But we can't do that as humans. So the products can be made in a timely manner when you have an enzyme around. Enzymes work by lowering the activation energy, which is the hill that must be first overcome, and that takes energy input. So it requires energy to get up the hill, and then we have the potential energy to run the kinetics. The hill, if it's the barrier between starting materials and their chemical products, then the enzymes lower that hill and will regulate metabolic activity. Enzyme proteins are very specialized and usually only catalyze one reaction. This is due to a requirement called a substrate. In figure 3.1, you can see that there are two different types of energy exchanges. One where energy is released, where reactants roll downhill to products and energy is released. That's an exergonic reaction. And the alternative is where energy is required. So the reactants are at a low energy state and the products are at a higher energy state. So energy has to be put into the system in order for products to be produced. So the enzymes help flatten the hill. In figure 3.3, we see a similar diagram in your book where a large hill is going to require a lot more energy than a smaller hill. Enzymes can also help regulate the rate of a reaction. When you think of an enzyme working with a substrate, think of the substrate as a key and the enzyme as the lock. They must fit together exactly or the door won't open. So in general, the reaction is the enzyme plus the substrate on the left side of the equation. ES with brackets around it indicates the enzyme and substrate are together, so you've put the key in the lock now. And when you turn it, you can open the door or get a product. Notice on the right side of this equation, you get your enzyme back. So the limiting reagent in this equation is the substrate. Enzymes can be recycled. They are not used in the reaction. They participate without being used up. So you'll need to refer to table 3.1 and figures 3.6 through 3.7 for more details. And I'll include a video through one of your help with chapter 3 pages in the unit 1 area. That'll link you to some support for these concepts, and we'll put some of these molecules in motion where you can help visualize these reactions that are being diagrammed here. So here in figure 3.6, we can see how the enzyme up at the top at 12 o'clock with the indication of letter E is available for a proper substrate. Now, substrates have to fit into the activation site, so only the substrate with the right shape is going to work like a key. If the enzyme is the lock, they have to fit together. So the square substrate, substrate B, and the hexagon substrate D, they won't fit. Only the round purple spheres with the S are the substrate that will fit. And you have the ES complex, like we saw in the reaction in the previous slide. The substrate down here at 6 o'clock gets converted to product. You see the little triangle with the P on it. The product then leaves the enzyme and goes off and does its work or participates in another chemical reaction, and the enzyme is then recycled and can be used again in another round of reactions. Let's switch gears now and look at our universal energy source called ATP. The food we eat and the oxygen we breathe are used to make this all-important nucleotide used in the transfer of energy. Shown in figure 3.12, the formation of ATP is a phosphorylation reaction where the terminal phosphate group is added to the ADP, forming ATP. So adenine diphosphate undergoes a phosphorylation reaction and becomes adenine triphosphate. The structure of ATP is shown here in figure 3.12, and we see that it contains a sugar, a 5-carbon sugar called a ribose, and it has linked to it a phosphate group and adenosine. Okay, so the business end of ATP is not necessarily the nucleoside, it is the phosphate groups. It is the triple phosphate linkage on the right of this molecule. So this is a very important form of available energy when those phosphate bonds are broken. So you have the oxygen, phosphate, oxygen, phosphate. Oxygen we know is very reactive. When those PO bonds are broken, you can release about 7.2 kcals of energy that's available for the cell to use to do work. So ATP is considered a universal energy form. Human metabolism can be summed up in 3 enzymatically controlled cellular steps. The first being glycolysis, which is an oxidative burn of sugar, and sugars are the preferred energy source. Then the Krebs cycle. The Krebs cycle is an intermediate and necessary preparatory step for electron carriers to be used in step 3, aerobic respiration. This is where oxygen is used as the terminal electron acceptor in the ultimate redox reaction to create continuous and large amounts of ATP. Please watch the provided videos, and I will resume this lecture on Monday, June 6, where we will continue looking at the details of glycolysis, Krebs, and aerobic respiration.